on to the third picture. It's not exactly like local.
Yeah.
But that's people. So also a very warm welcome to our third speaker this morning, Associate Professor Yusuke Morimoto, also from FITEC. He will be talking to us on signal transduction and cell motility for microorganisms. So he's also from FITEC, Department of Physics and Information Technology. His PhD in physics from Osaka. So, not physics.
No. Physics.
Not physics.
Biophysics.
Biophysics. Okay, physics and biology. So super clever. Okay. And his research interests include biophysics, molecular motors, signal transduction, intercellular pH, genome, fluorescence, microscopy, and cell migration. So I think he's going to have some pretty interesting technology to tell us about. And of course, like his colleagues, published a lot of very good papers in good journals. And welcome, and I hand over to you to present your work.
Thank you for the introduction. First, I'm glad to say given the chance to study in Europe. Oh, sorry. Yeah, today I will talk about the signal transduction and cell motility in microorganisms. That is dynamics of microorganisms. These are topics. At first, I talk about the strand mode, and next I talk about the bacterial studies. As you know, our bodies are composed of so many cells. Some biological techniques describe that the cell is one smallest unit of the living system. But in fact, there are many things in living cell, such as small organella and proteins and lipids. And another smaller one is ions. Living cells have multiple ions, and these ions concentrations maintain keep as constant concentration by homeostasis. These numbers are essential for several functions. Now we can measure and estimate these numbers in living cells using fluorescence techniques. There are many fluorescence probes developed, and microscopic techniques are developed. So now we can measure such as voltage and viscosity in living cells, and such as pH, ion concentration, and cell pressure, and intercellular temperature using fluorescence technique. My main technique is these fluorescence techniques. To use these techniques, we are studying the soil organism, soil slime mold, Dictyostelium. Dictyostelium has unique life cycle, including unicellular phase and multicellular phase. They live, normally live as a unicellular in soil region. And when they are starved, they aggregate and form a multicellular body within 1 day. So this is good model organism to study the cell development and cell aggregation. In cell aggregation phase, they use chemotaxis to cycle B uh, produced by themselves. Recently, we established the method to visualize these chemotaxis cycle signaling relay like this movie. This movie is macroscopic images. This scale by 1 millimeter, and these small green dots show each cell. The cell-to-cell signaling is uh, transmit like these spiral waves. The, from unicellular phase to multicellular phase, when we observe this cyclic relay, at first, this periodic signaling was observed, but after they formed the multicellular body of this, and here, this periodic signal is disappeared from this point. This means that they change the signaling the pattern in between unicellular phase and multicellular phase. And in unicellular phase, they use periodic signal, but multicellular phase, they don't use periodic signals. And this is interesting point because unicellular and multicellular use another other similar system. This result caused one thing because in multicellular bodies of Dictyostelium, what is acting as a signal? So the next, we focused on the calcium ion signals because calcium ion signal also oscillated in the stem cells when they aggregate to form a multicellular body. In multicellular body, when we observe the calcium signals, there are sometimes calcium firing was observed. This calcium spike is followed with the multicellular proton or motility. But this calcium firing is occasionally occur. So another experiment show that this is maybe by the mechanical stimulation because when multicellular body is working on the other plate, when they attach to the other plates, they show this calcium spiking. When I actively attach the plastic stick to the this multicellular body, they show a clear calcium spike like this. This result clearly shows that this calcium firing is caused by mechanism. This system is conserved in our human signaling and bacterial cell signaling. So this is very important signaling system for organisms. But when we want to know the molecular mechanism, this is a bit difficult point. In unicellular phase, not our group, another group showed that the piezo homolog channel works in unicellular cells for the mechanical stimulation. But when we observe these with piezo homolog deficient strain in multicellular bodies, these clear deficiency is not observed. This means that piezo homologs does not work in multicellular bodies. And another candidate pathway is calcium pathway from ER. When we checked the ER channels, the ER this when we deficient this pathway, the efficiency of the stimulus response was decreased. And curative agent and this mutant combination shows a clear no response. This means although the molecular mechanism differs between unicellular and multicellular cells, but the signal are the same calcium ions. This is interesting from evolutionary viewpoint. Like this, we are show a clear cell mechanism using fluorescence technique, like the cyclic signaling and calcium signaling. Now we are using this in microorganisms, but these techniques also used for other organisms. Next, I talk about another topics about our bacterial studies. Such as E. coli and Salmonella bacteria have 5 to 6 flagella, and they rotated flagella, and they can swim in liquid environment. Because at the base of flagella, like this rotary motor is embedded in the cell membrane. This looks very similar to artificial rotary motor, and the speed is also similar to artificial motor. And the rotational speed is almost 400 hertz. This means 400 rotation per second. And they rotate this motor bidirectionally, both sides direction shows. And the energy source is the proton multiforce, means the cell membrane potential and proton quotient. And so this means energy efficiency is very good, better than artificial motor. When we observe at the base of flagella, there the rotation motor. When at first ring structure is constructed in a membrane of bacteria, and after constructing the ring structure, at the center of ring, there's export apparatus is composed. Using this export apparatus, the proteins separated from inner cell to the outside of the cells. At the outside the cells, the protein is like this, like a Lego blocks. Protein is constructed axial structure like a filament. One filament is composed of about 100 to 10,000 proteins. They make about 1 to 10 micrometer version. This means cell size is 1 micrometer, so it's 10 times longer than their body. To achieve this, they have the protein specific sepsis secretion system. This apparatus is at the center of this motor. This secretion system using as driving energy source as These driving energy sources are proton multiples and hydrolysis ATP. The inner proteins transported from the cytoplasm are delivered to the outside of the cell, so transport it. Because this we can have the a panel inside of the plant. Like this, main topics of bacterial flagella is one is the rotational mechanism, and another one is the flagella construction mechanism, that is protein secretion mechanism. The important point is both energy source is proton multiples. So we want to know and we want to measure the proton flow in the motor. So indirectly, we to measure the this proton flow, we are using the GFP. Because original jellyfish GFP has high pH sensitivity. Because GFP has a chromophore, and this chromophore have the main two situations. And one is a protonation state, and one is a deprotonation state. Each state, they have this state, so the this chromophore is very sensitive to pH. The one is one GFP mutant is very sensitive to the pH, and it is used for the pH sensor. I also using this fluorine. This fluorine spectrum is changed depending on the surrounding pH. So the using this approach, we can measure the and we can estimate the intercellular pH of the living cells. We thought that if we use this probe to the motor, we can get the local pH around the motor. But at first, this quantitative we screened some mutant and we get the mutant and increase stability of score. And stability increasing improves image of using some present using this score. And to get shown of these probes, we use cryo-electron microscopy. The single particle analysis using cryo-electron microscopy showed that this probe is inside of the model. This probe is on. This means this floating probe was located inside the model. This means on the side of the export apparatus. So maybe this probe can measure the proton flow in the export apparatus. We can detect this probe's fluorescence as a fluorescence spot in the bacterial cells, and we analyze as the image processing and which calibration using purified proteins. And from these, we can estimate global pH. When you local pH with some deficient strains, when this ring structure is deleted, the local pH is a bit higher than wild type. Also, this exportability is deleted. This deletion also pH increase a bit. Maybe this means the export apparatus function is correlated with local pH changes. Another point is this mutant can make protein. This means this mutant can secrete proteins, but it is deleted. So the construction efficiency has decreased. This mutant also shows the higher local pH. So this means efficient protein transport by coordinated use of ATP hydrolysis and proton molecules. Maybe some chaperone proteins recruit to this state, and recruited proteins are segregated using proton molecules. We are measuring pH, but another ion activity is also measured. Years ago, we using mutants, we discovered that this secretion system is also use sodium ions. So depending depending on the situation, they use also sodium ions, not only protons. Recently, we discovered that this secretion system is works depending on the membrane voltage. When we increase the voltage, membrane voltage, membrane voltage, protein secretion level is also increased. So maybe the gate protein is gate protein shows the conformation changes depending on the membrane voltage and secretion mode of the change. From these studies, we discovered another bacterial single cells. They are small pH gradients. Near the cytoplasmic membrane, the pH is about 0.3 higher than throughout the cytoplasm. Near the membrane, there is a small pH gradient. This is maybe the very interesting point for cell physiological systems. Now we are studying what mean this mean what with cells. Next, I'll talk about another bacteria. This is spirochete bacteria. This causes in human and some animals. This shows motility and like this spiraling spiraling dynamics. Recently, we can isolate new species of spirochete, and these have interesting function. This is the photoactivated motilities. When we irradiate it by light, they activate the motility. With screening the mutant, we discovered that this is caused by the photoactivate cyclic suitabase is caused this function. This is almost new protein, so that there are no knowledge about the structure. So we using AlphaFold 2, we predict the structure and estimates the function. And we can transfer this to another bacteria. In coli, and this system also works in coli. Maybe this protein can use optogenetic tools and also in eukaryotic cells. These is achieved by many equipment in our apparatus. These are common equipment in our faculty, like this prior microscopy, supervision microscopy. This is based on the proton microscopy. Now we can use this equipment so we can achieve results. This is last slide. Thank you for listening. That's all.
Really love those videos. Very beautiful. Biology is very beautiful, isn't it? So I'm opening the floor for questions.
I'm just very interested on the Bacterial project. And you design a little line, how do you capture?
Sorry, this problem is developed by entrepreneurs. Then we go transfer to our cell system.
So your sensor,
This sensor is based on the TFP.
not on the flow, on the technical.
This is the calcium, sorry, calcium sensor based on the TFP. Yeah, TFP, this group has the TFP, TFP region and calcium sensor region. Then if calcium ion attached to this sensor, the fluorescence of TFP is changed.
Oh, good.
Yeah.
Thank you.
This sensor is almost a huge system.
Thank you for asking. It's all very new to another class. I'm also interested that the flagella structure using sodium ions, because with some bacteria they do use this sodium ATP as well, right? So it's, are there any other ions that can be used?
Yeah. Some groups shows that lithium ion or calcium ion. So maybe depending on the living environment, in ocean bacteria, they use sodium ion mainly, and in our soil environment, they mainly use just protons. Right. So depending on the environment.
So have you looked at the flagella in any of the bacteria that are specialized to pH environments?
Yes.
And is it similar or?
I think similar, but no one knows now. So our group also want to know
We have a question.
There was something as a bacteria that when you have a light on. Yes, fine.
Yeah.
So when you turn on the light, it was moving.
Yes.
Oh,
And they just come back.
oh,
These proteins conserved in soil bacteria. So now soil bacteria. So now we are thinking that when they normally they live in under the soil. So when they appear to the under the soil, they move to the lower portion and in the soil. So maybe they when they graduated by light and they move to the another.
so you generate improvement that we very that.
Yeah, no, we can do that.
But it's still very different from the even when you turn it on the light, but it's not
Yeah.
You find soil growth so that the actually stay in depth.
We made some mutants, and it's non-response mutant show back and
Like just it's like out. We do job.
when we discover we also very yes.
So actually, the movement uses is very energy consuming, right, to drive this movement.
Maybe using the adjacent.
So automatic process for products of respiration.
Yes.
So we then would use a lot of energy for
Yes.
this.
But there are a lot of this.
Is there any applications of this for alternative or renewable energy?
Yeah, we want to do
Right. Can you tell us a bit about your ideas, or it's still secret? Okay. Because it's very exciting when we look at biology. We always think of petrochemicals, glucose and ethanol. There's all these alternate energy systems that microbes have been using for millions of years. So it's very interesting and very kind of thing that there's a lot of protons. So that's that's a good source.
There was one slide in this there. It works very well. There was this two, one is yellow and another one is that works very well for multicellular system, but it's that very what is your big one? On how the one you saw? Yeah, this one. What is your expert opinion on this? How to address this? Because a lot of us work on the eukaryotic system with multicellular. And the benefit is the one that is a unicellular. Where do you see this technology taking towards the tackle multicellular situation?
Yeah. It's a very revolutionary point. The interesting point is similar to same. Both use calcium ion, but they use different gene. That means a different protein use to achieve this signaling systems. So maybe it's teaching when they change from unicellular to multicellular forms, gene special whether it's completely different. So in different situations to achieve same signals, maybe they change the molecular mechanism also have to change. So I don't have a clear answer about it.
Why am I watching it?
Yeah.
Time for so in life now, what is this technology like? The damage or the at a very early stage, really advanced stage, whatever it is. Yeah, that's very nice. But that is what is needed for a medical point of view. Because the kids are growing up because of the radiation, because of the environment. Just hope that your technology goes together. That's what you think.
Any more questions or comments? Okay, just be reminded of the question or two.
I was just maybe one like I think that you mentioned that the area closer to the membrane has higher reach.
Yes.
What do you think that would imply? I mean, in terms Why do you think it's important to have it? How does that affect the that you come here?
Yeah,
Potential like
well, one point is the bacteria you is a motility and so bacterial model is proton molecules. This proton flow is always from outside to inside, but in some high pH like high pH high pH environment like bacteria in maybe around pH 11. In these conditions, outside of pH is higher. This means that proton concentration is very low outside, but they hold the proton from outside to inside. So someone says that, well, maybe they can reach the gradient only around the cell membrane. So this method is also maybe correlated this
in the cell, everything goes and then grow when you come to the wider.
Yeah. Maybe there are some issues. Yeah.
Right. Like this is a different here. So like when you say when the light spot on something, so like there's a bacteria move for a certain direction to avoid the light or the light.
Not
Not what the light. Why so then the one thing is by because it's in your the chips that you is so right is the same chip. Let's say people are so the same. That's the system the chips that you may be about here.
yeah, I want to but we cannot also so that equal so the resources is happened by the photo realization, but what is not changed. So another similarly passwords for in the spiral.
So even you put the chips people are able to
Sounds on your back.
that's
That's the other thing about biology. It's complicated. A lot of redundancy.
The viruses are really very complex people.
Yeah.
A lot of diseases quite very practical. And geneticists keeps evidence with you.
But I think an important point that comes out is that sometimes when we learn basic biology, we learn it very simplistically. Is this or is that? As eukaryote, prokaryote, and we don't learn the actual complexity of it. And it can lead us to not thinking broadly.
That's why you are highly important to move Jenny from the point.
Yes,
Because we are all connected as one.
yes.
In a ecosystem. You matter the fact of point you cannot learn the teaching of the system. There's all.
So I think we can open to questions for any of our speakers. Maybe some of you have been thinking about, oh, I missed my chance because the other professors, did anything occur that you worked at? Never stop asking questions. Then you have to retire people stop asking questions. Or you ask different questions.
I think the students make them really.
So any questions for any of our speakers?
The professor here You see the bright, the pink, and like.
So if there is no more questions, please join me in thanking all of our speakers for the excellent presentation. Come back to
So much. Everyone for paying attention, and thank you for our presenters for willing to share the knowledge with us today.